diffusion, and in this facilitated diffusion, mediated transport, and in mediated transport we shall discuss about facilitated diffusion as well as the process of active transport. Can you see the slides? Okay, facilitated diffusion is it is basically what we can call it as an improper term because this process actually it does not involve the process of diffusion, but at the end of the process, when we look at the end product, the end product of both diffusion and facilitated diffusion, they are more or less same. And in this facilitated diffusion process, the net flux of an uncharged molecule. Uncharged means that molecule is having no positive or negative charge across a membrane, that will always be from higher concentration to lower concentration, and it will continue until unless the concentration of the solute that becomes equal on the two sides of the membrane, that is called diffusion equilibrium. So in case of facilitated diffusion, equal number of the molecules they are binding to the surface of the transporter protein at the outer surface, and similarly equal number of the molecules, they are also moving to the inner side of the membrane from the same transporter protein. So neither diffusion nor facilitated diffusion, as both of these processes, they are down the concentration gradient. Mean both these processes, they involve the movement from higher concentration of the molecules to their lower concentration. That's why there is no involvement of the energy, and that's why these two processes they are always with the concentration gradient or down concentration gradient. And most important facilitated diffusion system, when we talk about the facilitated diffusion in our body, the most important facilitated diffusion process in our body is the movement of the glucose across the membrane. As all of you know, that glucose is transported into the blood and into the various cells with the help of glucose transporters, and those glucose transporters are GLUT4 molecule and GLUT3, GLUT4. These are some glucose transporters which are actually involved in the transport of the glucose molecule across the membrane of certain blood vessels and certain cells. So when these glucose transporter, which we have called as GLUT transporter, when these transporters are absent, cell will be impermeable to the glucose, and hence glucose molecule, they will not be able to move across the membrane, because this is large molecule, this is polar molecule. It cannot pass through the lipid bilayer of the membrane. That's why transport of the glucose is facilitated with the help of specialized or the specific transporter, and those are called glucose transporter or GLUT transporter. So as a result of facilitated diffusion, if we talk about the glucose concentration, apparently it seems that there should be equal concentration of the glucose on the two sides of the membrane, but as we have discussed in very first lecture, that when we talk about the extracellular concentration of the solute is normally present at very high concentration on the extracellular side. So that's why it will not have an obvious effect when some molecules they have moved from extracellular side to the inside of the cell, whereas when we talk about on the inner side of the cell, it will have definite effect on the inside of the membrane. So losing few glucose molecules from the extracellular side will not have an obvious effect on the extracellular concentration of the glucose, because glucose once it enters into the cell, it is converted into the glucose 6 phosphate. When it enters, it is converted into the glucose phosphate. Can you see the slides?
Yes, sir.
Okay. As all of you are familiar with the process of glycolysis, that once the glucose enters into the cell, it is converted into the glucose phosphate, and a cyclic process is carried out inside the cell, which is called glycolysis. So that's why glucose concentration, it can't be equal on both sides of the membrane, because the glucose molecules which are entering inside the cells, they are immediately they are metabolized and they are converted into glucose 6 phosphate. So intracellular glucose concentration, it will always it will be lower as compared to the extracellular concentration. And although there will be continuous flux of the glucose molecule inside the cell, but all those glucose molecules which are being transported from the extracellular side to the intracellular side, those glucose molecules, they are basically metabolized and they are converted into glucose 6 phosphate. Okay. Okay. Uh, there are different transporters which are involved in the movement of the glucose across the membrane. As I have told you that GLUT2 is one of them, GLUT4 is another molecule. These are the glucose transporter which transport the glucose molecule from the extracellular side of the membrane to the intracellular side. And as these transporter molecules are the carrier molecules, they are basically protein in nature, and these proteins are encoded by the genes. As all of you know that proteins are formed by the genes. They are coded by different sort of the genes, and those genes are expressed in different types of the cells. So transporter, when we talk about their affinity for binding with the glucose molecule, that is also different in different types of the transporter molecule, and their the maximum rate of the transport will be achieved when the transporter are these glucose carrier proteins or the GLUT molecules. They are completely saturated, mean there is no more binding site available for further binding of the glucose to these GLUT transporter are these transporter molecules. So at that particular time, the maximum rate of the transport that will be achieved by the cell, and the transport activity that can be changed or that can be modulated by the help of certain stimulus or with the help of certain signals, chemical signals. For example, a hormone, insulin. As all of you know that insulin can lower the blood glucose concentration, or the concentration of the glucose which is freely present inside of the cell. So at that point, when there is maximum rate of the transport of the glucose across the membrane, certain types of the chemicals and hormones, and specifically insulin, can modulate or change the glucose concentration inside the cell. So glucose, it enters into all types of the cells with the help of these transporters, which are called GLUT molecules, GLUT2, GLUT4 molecules. And insulin, as I have told you, that insulin actually affects some specific type of the glucose transporter, and those glucose transporters which are present in the or expressed in the muscles and the adipose tissues, insulin basically affect those glucose transporters. As all of you know that glucose is converted into free glucose is converted into the glycogen, and this glycogen is stored in our muscles as well as in our adipose tissues. So insulin will actually affect those transporters which are expressed in muscle cells as well as the adipose tissues of the body. And as all of you know, that insulin will increase the glucose of the will increase the glucose transporters in the membrane, and hence the rate of the glucose movement that will also be increased with the help of the insulin molecule. So here, so this type of the facilitated or the mediated transport is called as facilitated diffusion, and primarily the glucose is transported from the extracellular side to the intracellular side of the various type of the cells with the help of facilitated diffusion or the mediated transport. And the transporter proteins which are in glucose transport, they are called GLUT molecules. They are either GLUT2 or GLUT4 molecules. The second type of the mediated transport system, that is called as active transport. Active transport, as all of you know, that it differs from the simple diffusion or the facilitated diffusion, in the sense that it will involve the expenditure of the energy, and the molecules will always move from lower concentration to higher concentration, and that is what we can call it as against concentration gradient. So active transport, it is carried out in a similar fashion as like that of facilitated diffusion, that in this process specialized type of the proteins which are called carrier protein, they will be required. So here you can see in this figure that in case of diffusion, or in case of diffusion, there is simple movement from higher concentration to the lower concentration across a membrane that is permeable to these molecules, whereas in case of facilitated diffusion, again there is movement of the molecules from higher concentration to their lower concentration, but in this process there is involvement of a specific intermediate protein which is called as carrier protein. And when we talk about the third type of the second type of the mediated transport system, which is called as active transport. In case of active transport, the molecules are moving in opposite direction. I mean, they are moving from their lower concentration to their higher concentration. This is movement, uphill movement, or we can also call it as movement against the concentration gradient, and this process will always require the expenditure of the energy. So these transporter which are involved in active transport, they move the substances uphill, mean from lower concentration to higher concentration. That's why these carrier proteins or these transporters, or sometimes they can also be called as pumps. As you can commonly observe that pump is something which actually move the substances in uphill direction, meaning lower concentration to higher concentration. For example, when we talk about the heart as our pumping organ, it means heart will transport blood from lower part, or it will transport the blood to the upper part of the body, so where it is actually acting as pump. So because of the nature of the action of these carrier proteins which are mediating the process of active transport, and they are actually moving the molecules from lower concentration to higher concentration, or uphill movement. You can simply understand this is as it is an uphill movement where the molecules are moving from lower concentration to higher concentration. That's why these are called as pumps. In case of active transport, as the carrier proteins or the mediated proteins, the transporter proteins which we have discussed in facilitated diffusion. Similarly, the carrier proteins, which we have also called them as pumps, they also show the specificity for binding with the specific type of the solute particle, and they also show the property of the saturation. Mean at a specific time, the binding site of these carrier proteins or the transporter protein will be completely occupied, and no more binding site will be available for further binding of the solute particle to this transporter protein. So this point is called as saturation. So these transporter proteins which are involved in active transport mechanism, they also show the specificity for binding to specific type of the transported solute, as well as they also show the saturation. And similarly, when we talk about the flux via the transporter protein, the flux will only will be maximum, or it will achieve the maximum rate when all the binding site of these transporter protein they are occupied. Mean there is complete saturation of these transporter protein, and the net movement will be from lower to higher concentration, and higher steady state concentration on one side of the membrane. It can always be achieved with the help of input of energy. Mean energy will be continuously required to carry out the process of active transport, because this is uphill movement and it will always require the expenditure of energy. So how this energy is actually provided, how this energy can be harvested, and what will be the ultimate function of this energy. This energy, the first function of this energy is the changing of the affinity of the binding site. Mean when the energy will be provided, the transporter protein will show high affinity for binding to the solute particles. So when this transporter protein will show high affinity, the solute particle will bind to this transporter protein at higher rate, and they will be facing to the one side of the membrane as compared to the other side. Meaning when the carrier proteins or the transporter proteins, they are showing the upper configuration towards the one side, it means they are showing the high affinity for binding, and at the other side they will be at that particular time they will be showing the low affinity. The second important function of this energy is that it will alter the rate at which binding site of the transporter is shifted from one surface to the other. Mean from open configuration to the closed configuration. When the transporter proteins are in open configuration, it means they are showing the high capacity or high affinity for binding to the solute particle. And when these transporter protein they are showing the closed configuration, it means they are having very low affinity for binding with the solute particles. So the rate or the speed is also altered by this energy. When the energy is provided, the speed of these, the binding site, the availability of the binding site will be increased as a result of input of the energy. And the third function of this energy is that in order to move molecules from lower concentration to higher concentration, energy will be required. It will be continuously required to carry out this process. So these are the important functions of the energy. That's why we can say that energy is very much required to carry out the process of active transport. So active transport, it should be simultaneously, it should be provided, or it should be coupled with the flow of energy from higher energy level to lower energy level. Mean energy should be provided from any specific type of the energy source to carry out the active transport. And two means of the energy coupling of the energy to the transporter, they are known. One is direct use of the ATP. When the energy is directly utilized by the cells, by the molecules, by the transporter protein in the form of the ATP, that process is called active transport. And sometimes the use of an ion concentration difference across a membrane, and this ion concentration difference will drive the process, and that will be called as secondary active transport. So this active transport is further, it is divided into two types. One is called primary active transport, and second type is called as secondary active transport. In case of primary active transport, as we have discussed, that it actually involves the direct use of the energy in the form of the ATP. So hydrolysis of ATP by transporter, it will provide the energy. As all of you know that when the energy is utilized, the ATP will be converted into ADP, adenosine diphosphate, plus inorganic phosphate. And this hydrolysis of the ATP is is accomplished, is carried out with the help of an enzyme which is called ATPase. ATPase is an enzyme which specifically hydrolyzes the ATP into ADP plus inorganic phosphate. So transporter proteins are actually having the ATPase activity. They are showing the ATPase activity. I mean, they have the ability to convert this ATP into ADP plus inorganic phosphate, which is called ATP hydrolysis. And when these proteins are carrying this ATP hydrolysis activity, it means these transporter protein they should have an enzyme, and that enzyme is having ATPase activity. It will break down the ATP and it will phosphorylate itself. Mean the inorganic phosphate that has been released from the ATP, that inorganic phosphate will be utilized for the phosphorylation, and hence these transporter protein, they will phosphorylate their surfaces. And phosphorylation of the transporter protein, it is actually a covalent modulation or covalent modification. When the phosphate is added to these transporter protein, their affinity will be changed and they will show a high affinity to the solute particles. Means the binding site of these transporter protein for the solute, they will be available now, and these transporter protein will show the high affinity. So in upcoming slide, we will see the sequence of the event which are leading to the active transport from lower concentration to higher concentration. So here you can see that in this process ATP is directly involved, and in the first step this ATP is hydrolyzed and it is converted into ADP plus inorganic phosphate. That inorganic phosphate, it will be added to the surface of this transporter protein, and hence their phosphorylation will be accomplished. When the phosphate is added to the transporter protein, their affinity to these solute particles will be increased. Their affinity for binding to the solute particle will be increased, and they will show high affinity. And once they will show high affinity, the solute will bind to these transporter protein, and ultimately the solute will be transferred, will be shifted towards the inner side. And once the solute particles they have been transferred to the inner side, this inorganic phosphate will be released from this transporter protein, and it will again bind with the ADP, and it will convert this ADP plus inorganic phosphate into ATP. This ATP is now ready to facilitate the movement of other solute particle by repeating the mechanism. I mean, by hydrolyzing itself into ADP plus inorganic phosphate. That phosphate will again inorganic phosphate will bind to this transporter protein. It will phosphorylate the transporter protein. When this transporter protein is phosphorylated, the binding sites will be available and solute will easily bind. This transporter protein will show high affinity. So this is these are the sequence of the events which ultimately result in movement of the molecules from their lower concentration to the higher concentration. This is primary active transport model, and binding sites, their affinity is changed once the phosphate group is added to this transporter protein, and this affinity is changed by phosphorylation and dephosphorylation. When this is dephosphorylated, now at this point, this transporter protein will show very low affinity for binding to the solute particle. But when this is again phosphorylated, this transporter protein, when it is again phosphorylated, it will show high affinity, and the solute particles will easily bind to this transporter protein. So this is active transport model. So binding site of the transported solute, initially that is exposed to the extracellular fluid, and it will show high affinity because of the phosphorylation, because the protein has been phosphorylated on its intracellular surface. And remember that the protein will not be phosphorylated on the outer side. The protein will be phosphorylated in the inner side of the membrane. It means this ATP is being provided from inside of the cell. It is not being provided from the outer side of the cell. It is being provided from the inner side of the cell. Mean any energy source. For example, as all of you know that mitochondria is power of the house of the energy. So this ATP that might have been provided by the mitochondria, which is present in the cytoplasm or in the inner side of the cell. So once the transporter protein on its intracellular side, it is phosphorylated with the inorganic phosphate as a result of hydrolysis of ATP, and this protein is having ATPase activity. When it phosphorylates this ATP, simultaneously the affinity of this transporter protein for this transported solute, it will be increased, and this solute will easily bind to the transporter protein. So this is first event, that phosphorylation of the carrier protein and the transporter protein will increase its affinity. Once this phosphorylation process will occur in the cell, and it will always occur on the intracellular side of the cell. And now the conformational changes will be there. Now the conformational changes will be there, and protein, this transporter protein, they will be in the open configuration. The transported solute in the extracellular fluid, it will bind to the high energy binding site. Now means that the transporter protein, they are showing the high affinity, and the transported solute will easily bind to this carrier protein, and there will be thermal oscillations. It will repeatedly expose the binding site to one side of the membrane, then to the other, and sometimes this is not dependent upon the phosphorylation process. It is not strictly dependent, as we have previously discussed, that these carrier protein or the transporter protein, they remain in a sort of oscillation. Mean they sometimes they remain in this configuration, which is open configuration, and some other time they will be in the closed configuration, and these shapes, they are repeated time and again. This is called oscillation, and these movement, these thermal oscillation, they are not totally dependent upon the phosphorylation mechanism. Mean without this phosphorylation, here you can see that this carrier protein or transporter protein has again assumed this shape. It has again assumed this shape, and here no phosphorylation is there. So we can say that this open and closed configuration of this transporter protein, it is showing the thermal oscillation. They are repeatedly exposing their binding site, either on one side of the cell or on the other side, and this change in configuration or conformation, it is not totally dependent upon phosphorylation. Mean without phosphorylation, this transporter protein, they can assume the open or closed configuration. And once the phosphor group is removed from the transporter, the affinity of the carrier protein or the transporter protein to the solute particle will automatically, it will be decreased. And hence, when the affinity of the binding site will decrease, the transported solute will be released from the transporter protein, and it will be released to the intracellular side. Now when the phosphate is removed, it means there is now the affinity for binding of this solute to this carrier protein that has decreased, and ultimately this solute particle will be shifted, will be transferred to the intracellular side of the cell, and it is basically a conformational change which will again permit the phosphorylation, and hence the cycle will be repeated, as you can see in this example, that once the transported solute has been transported towards the intracellular side, again the shape of this transporter protein will be changed, and these repeated changes will facilitate the movement of the further molecule to the intracellular side of the cell. So this will lead to the movement from lower to higher concentration. Mean up, there will be uphill movement, and flow of the solute. It will continue unless until the solute on the two sides of the membrane, the solute concentration that is equal, and more solute will bind to high affinity state at extracellular surface of the membrane, as compared to the low affinity site which is present on the intracellular surface. And when the more solute will bind, definitely it will result in movement of the more solute towards the inner side, in then out, when the transporter basically oscillate after its transporter protein, it show the repeated changes, conformation changes in their shape. So major primary active transport proteins which are present in the cell, they are called as sodium potassium ATPase. One of them is sodium potassium ATPase, and similarly we have calcium ATPase, we have hydrogen ATPase, and we have hydrogen potassium ATPase. Why we also add the word ATPase mean these transporter protein, they have the ability to hydrolyze, to break down the ATP, and hence this property is called ATPase property or ATPase activity. Sodium potassium ATPase, it is present in almost all the plasma membrane, and pumping activity of this active transport protein, it will ultimately lead to the distribution of the intracellular potassium and low intracellular sodium. Mean it will actually distribute the sodium and potassium unequally to the two sides of the membrane. I mean, potassium will be high towards the intracellular side, and sodium will be high towards the extracellular side relative to their extracellular concentration. So for each molecule of the ATP that is actually hydrolyzed, here you can see that one molecule of the ATP is hydrolyzed. The transporter will move 3 sodium ion outside of the cell and 2 potassium. Mean when the ATP is hydrolyzed, these sodium are already they are having low concentration on the inner side of the membrane. So when one single ATP molecule is hydrolyzed, it will result in movement of 3 sodium from inside to the outside, and 2 potassium from outside to the inside. And when we talk about, this is the concentration of the sodium and potassium. The extracellular concentration of the sodium is 145 millimole, and potassium is this is 4 millimolar, in some cases it is 5 millimole. And when we talk about the intracellular concentration, sodium is having 15 millimolar concentration towards the intracellular side of the cell, whereas the potassium is having 150 millimole. It means potassium is highly concentrated on the intracellular side, and sodium is highly concentrated on the extracellular side. So when the active transport will be operative, it means the potassium will move from their lower concentration to higher concentration, and sodium will move from their lower concentration to their higher concentration. This is what we can call it as active transport mechanism. Okay, so here you can see that the primary active transport of the sodium and potassium ion, it will always be in opposite direction, and it will always be in against the concentration gradient. Means sodium is moving from lower concentration to higher concentration, from inside to the outside, and potassium is moving from lower concentration to the higher concentration, and it is also moving from outside to the inside. So this is active transport of the sodium potassium, and it is carried out with the help of sodium potassium ATPase. This is actually the protein which is carrier protein, which is transporter protein, and it is having the sodium potassium ATPase activity. It will hydrolyze the ATP, and single ATP hydrolysis will result in movement of 3 sodium from inside to the outside, and movement of 2 potassium from outside to the inside. So it will result in net transfers of positive charges to the outside of the cell. As 3 sodium will move, it means sodium is always already having higher concentration on the outer side, and when more sodium is being transported, the positive charges will increase on the extracellular side. And as you know that 3 sodium are moving, and in response of that 2 potassium are moving inside, so definitely there will be low positive charges on the inner side of the membrane as compared to the outer side of the membrane. So net transfer of the positive charges will be increased, and the transport process is not electrically neutral. This process is not electrically neutral. Neutral in the sense that there will be no equal number of the positive and negative charges on the both sides. As I have told you that when the 3 sodium will move from inside to the outside, and 2 potassium from outside to the inside, so definitely the number of the positive charges will be unequal now. Mean outer side of this cell will have high positive charges, higher concentration of the positively charged ion, and inner side will have lower concentration of the positively charged ion. So this is the what we can call it as net result of the positive and negative I to the two sides of the membrane. The second type of the ATPase, as we have discussed about the sodium potassium ATPase, that it is present in almost all types of the cell. When we talk about the calcium ATPase is present in plasma membrane, as well as it is also present in membranes of certain organelles. For example, we have mitochondria, we have endoplasmic reticulum, and certain other organelles. They are also having their plasma membrane. So this calcium ATPase pump is not only present in the plasma membrane of the cell, but it is also present in the plasma membrane of the various organelles of the cell. So plasma membrane, when we talk about the plasma membrane, the direction of the active calcium transport will be from cytosol to the extracellular fluid. The calcium is highly concentrated on the outer side, and it is having low concentration in the inner side. And when there is active transport of the calcium, it means the calcium will be transported from inner side of the cell towards the extracellular fluid. And in organelles membrane, this is what we are talking about, the plasma membrane. When we talk about the organelles membrane, in organelles membrane, it is from cytosol organelles membrane. We will clear this point in upcoming lectures, where you will observe that calcium is having higher concentration in endoplasmic reticulum or the sarcoplasmic reticulum as compared to the free calcium which is present in the cytoplasm. So when we talk about the movement of the calcium across a plasma membrane, and this movement is mediated via the active transport mechanism. So in this case, in that case, the calcium will move from inner side of the membrane towards the outer side, mean from the cytosol or the cytoplasm towards the extracellular fluid. But when we talk about the movement of the calcium in the organelles, we will say that calcium is having lower concentration in the cytoplasm as compared to the calcium concentration in the various organelles, and specifically in the endoplasmic reticulum. So in that case, the calcium will move from cytoplasm, where it is having low concentration, towards the organelles lumina, the cavity of the organelles, where calcium is having higher concentration. So this active transport of the calcium out of the cytosol from lower concentration to higher concentration, it is accomplished via calcium ATPase pump. And this is the reason that cytosol of the most of the cells, that they are always having the low calcium concentration, about 107 mole per liter, as compared to the extracellular calcium concentration, which is 103 mole. I think there must be some sort of the error in this digit. It should be higher than this value, because cytosol is always having the lower concentration of the calcium. I will recheck this, and this extracellular side will have the higher concentration of the calcium, and it is 10,000 times it should be greater. The third type of the pump is hydrogen ATPase pump. This is plasma membrane and several organelles, membrane of the several organelles. They are also having this hydrogen ATPase pump, and specifically it is present in the inner mitochondrial membrane. As all of you know that mitochondria is having two double membranous structure. It is having an outer membrane and is having inner membrane. So this hydrogen ATPase pump is present in the inner membrane of the mitochondria, and it is also present in the lysosomal membrane. Certain lysosome, they are also having these hydrogen ATPase pump. And when we talk about the hydrogen ATPase pump present in the plasma membrane, it will move the hydrogen ion out of the cell. Hydrogen potassium ATPase is another type of the pump which is present in the plasma membrane of the acid secreting cell. As all of you know that acid secreting cells are present in our stomach as well as in our kidney, and it will move one hydrogen ion out of the cell, and it will move one potassium in for each molecule of the ATP hydrolyze. This hydrogen potassium ATPase pump, when it hydrolyze one ATP molecule, it will result in movement of one hydrogen ion from inside to the outer side, and at the same time simultaneously it will also facilitate the movement of one potassium from outside to the inside. So this pump is electrically neutral as compared to the other pumps, because there is no one positive ion is being transported from outer side to the inner side, and simultaneously another positive ion is being transferred from outer side to the inner side. So there will be no difference of the charges. Electrically this moment is neutral. Here again we have also discussed this slide, that here is extracellular concentration and intracellular concentration, and as I have told you that when a single ATP molecule is hydrolyzed, it will result in movement of 3 sodium ion from inside to the outer side and 2 potassium from outer side to the inner side. So hence in this mechanism there will be difference of the charges, mean membrane potential will also generate. The membrane will have more positive charges, this plasma membrane will have more positive charges on the inner side, and it will have more negative charges on the outer side of the membrane. This is all about for today. Okay ji. Any question?
Sir, last slide.
This one? Okay. This one or this one?
Yes, sir.
This one?
It's the next.
This one, hydrogen ATPase pump?
Yes, sir. It's the next.
इससे नेक्स्ट तो ये आ जाती है ना.
Yes, sir.
Okay, here we have discussed that concentration of the sodium is more on the extracellular side, and sodium is having low concentration on the intracellular side, and similarly potassium is having higher concentration on the intracellular side, and it is having lower concentration on the extracellular side. So what will happen with the help of sodium potassium ATPase pump? This circle is representing the sodium potassium ATPase pump. When this sodium potassium ATPase pump, it will hydrolyze a single ATP molecule and it will convert this ATP into ADP plus inorganic phosphate. That phosphate will phosphorylate this sodium potassium ATPase pump. It will facilitate the movement of sodium from its lower concentration to its higher concentration. This is lower concentration of sodium and this is higher concentration of sodium, and simultaneously the single ATP molecule, when it is hydrolyzed, it will result in movement of 3 sodium. 3 sodium will move from sodium lower concentration to sodium higher concentration, and similarly as potassium is already having lower concentration on the outer side and having higher concentration on the inner side, so when a single ATP molecule is hydrolyzed, it will result in movement of 2 potassium from outer side to the inner side, from lower concentration to higher concentration. So as 3 positive ions they are moving towards the outer side, and 2 positive ions they are moving towards the inner side, so definitely there will be imbalance of the positive ion, mean on the one side there will be more positive ion and on the other side there will be less positive ion, and then when there are less positive ion, it means there are more negative ion, so there will be separation of the charges that will also be present, mean outer side will have positive charges and inner side will have the negative charges. Okay, got my point?
Yes.
Okay, any other question please? No question?
सर ये सारे लेक्चर जो हैं वो हमें स्लाइड्स में मिल सकते हैं?
Yes, I will share the PDF of all these lectures with you.
जी सर.
Okay, ji. Thank you very much. Please stay connected.